right, here we go. So here's where we left off, right? This is the last thing we took notes on. I have like this huge long page down here. I'm not going to use it. I like to, when I start a new section, I like to start a new page. If you want to use it, go for it. Go for it. Again, these are your notes. So, if you got pages you want to use, you don't, however you want to do it, up to you. But I like to start a new page. I want to start a new day. And today, we are taking a look at DNA replication. It's a really important process. You have 37 trillion cells in your body, and they all have the exact same DNA. So it's a really important process that we can replicate our DNA. Whenever a mistake is made, we call it a mutation. Not all mutations are bad, but some really can be. So making sure we do a good job copying our DNA is a really, really important aspect of life. First of all, let's introduce the players. We're going to use a triangle to introduce our first guy here. This is what we call helicase. Helicase, and quite frankly, the name, the word, tells you everything you need to know about him. Don't believe me? Let's see what he does. A protein, how do we know it's a protein? It ends in ASC. Enzymes, proteins, end in ASC. So right there, we know we're working with a protein. And that protein unzips the double helix. That's helicase's job. So hela, kind of like a helix. So yeah, the name kind of tells us everything we need to know about them. Unzips the double helix, helicase. Next up, got this little tiny guy, little tiny circle. We're actually going to have quite a few of these little tiny circles. They're called SSBPs, which stands for I can say we just call them SSBPs. Stands for single-stranded binding proteins. Again, you're probably never going to say that combination of words ever again. You're welcome to just say SSBP, but that's what it stands for. And their job is to hold the strand open. Helicase unzips it. SSBPs keep it unzipped. Next up, here's a big shot for you. This one's called DNA polymerase. Again, most of what you need to know about this guy is right in the name if you know what to look for. Again, ACE, ASC tells you he's an enzyme, a protein. Polymerase, polymer. We just need to know which polymer does he make. There it is, DNA. DNA polymerase is a protein that adds nucleotides based on base pairing rules. Base pairing rules we've talked about. A pairs with T. C pairs with G. So the straight line letters A and T, I think with the capital letter versions, they're both made with straight lines. They go together. And C and G, the curved lined letters, they go together. So those are our base pairing rules, and DNA polymerase knows that. Unfortunately, he's got two weaknesses. Number one, his first weakness is that he can't start his own strand. He can only add on to an already existing strand. So that's a pretty big problem for him. Secondly, second weakness is that it can only attach nucleotides 5' to 3'. So perhaps you guys remember earlier this week I should say, we drew those diagrams of nucleotides, and I insisted on showing you the numbers. And I said these numbers may not matter right now. And they didn't seem to at the time. They didn't at the time. But now they do. Here's where and why they matter. DNA polymerase can only attach a 5' end to a 3' end. Let's take a closer look at that. So here's our sugar. Remember, he's got that elbow and then he's attached to a phosphate. Sticking off on this side is the base. But right now we don't care about him. He's not really factoring into any of this right now. So I'm not going to worry about drawing pentagon, hexagon, any of that stuff. Just going to call him a base. And then when we number this, again, this is number 1. Over here where the base is, 1'. It looks like an apostrophe. It's a prime symbol. I go 2', 3', 4', and then the elbow is 5'. So again, DNA polymerase can add nucleotides, but he can only add 5' to 3'. This he can do. He can add the 5' end of this one to the 3' end. Can do. This attachment can be made 5' to 3'. What he cannot do, draw another one up top up here. So what he is unable to do, I'm going to draw little X's so you know he's not able to do this, is he can't add them in that direction. He can't go 3' to 5'. Just doesn't work that way. So always remember going down 5 to 3, down 5 to 3. 5 to 3 is the proper direction. And again, right now, that may not seem like such a big deal, but it really kind of is. And I'll continue to show that to you the next few minutes here. Questions so far?
How do I PDF the seating charts?
Go to print.
Go to
Oh,
print.
they're document right now?
I have a meeting yet.
Oh.
Put in. So I got to make those, and I don't know how to do
Okay,
it.
generally you go to print,
print.
and then instead of picking a printer, you can convert, there's an option to convert to PDF.
Okay, that's what I'm doing.
Yep.
Okay. I got to get those in, like, Beth asked yesterday.
All right, questions so far? Next up, we get a square. And it's not really a square in real life, just the shape I'm using. This one is called Primace. And there's that ASC again. And technically, we're going to call him Primace for now, but technically, good thing to know is he is in RNA polymerase. So that kind of tells you what he does, kind of. He can and does he can start a new chain. So remember up here, DNA polymerase, one of his problems is that he can't start a new chain, but Primase, which is an RNA polymerase, does and can start a new chain. His weakness, yes, they all have a little weakness, don't they? Hey guys, you're a little larger than me and you're out of page. That's fine. Go to the next one. That's fine. That's fine. I've got one more thing to write, and then I'm going to the next page. So if you get there one line before me, not a big deal. Your notes. So his Primace's weakness, kind of in the name here, is that he adds RNA, not DNA. We're trying to duplicate DNA. We'll get to RNA next week. We're trying to duplicate DNA, so this is a pretty big weakness here, adding RNA nucleotides. Finally, we got ligase. I tried to make this one more of an oval. We've got the circle, it's RNA polymerase up here. This one I tried to make a little more of a circle. Ultimately, it doesn't matter. It's not really his real shape. I'm just trying to use different shapes to distinguish them when we diagram with them. So something of an oval. Ligase, why is he there? he is capable of joining DNA 3' to 5'. So he's got one job to do, kind of cure the weakness of DNA polymerase up here. Questions? Can you give us a quick pause here? Let you get caught up on this page. I'm about to flip it. So any questions on this page? And we need another minute or so. We're good to go. All right. Very cool. Let's get to the replication. Now that we've met the players, let's see how it works. Lunchtime for someone else. Hey, hopefully we're all kind of recognizing this shape, huh? And there's one of the players we just met. Remember the triangle? That's helicase. How about the small circles? Those are SSBPs. So remember, our strands of DNA are anti-parallel. They're running in opposite directions. So if the top strand here goes 3 to 5, the bottom strand would go 5 to 3. They're anti-parallel to one another. So this opening here is called the replication fork. It's just like a fork in the road, like a split. That's the replication fork. It's the split where we are going to start replicating. All right, so what we have happening is helicase comes in and lands at this site on the DNA strand that we call the origin of replication. Hopefully that title is kind of self-explanatory. Origin, like the beginning of, this is where we're starting our DNA replication. And helicase begins to unzip the DNA. It's very much like unzipping a jacket. You know, your jacket's got the two keys that are stuck together, and you start to unzip it. So that's kind of what we have happening here. different from a jacket. Each chromosome has an estimated 10 to 100,000 origin of replications. So it's not like we're just starting at the bottom of the DNA strand or at the top of the DNA strand. So very much unlike a zipper on a jacket. We're starting all over the place. And in fact, we're going to kind of diagram that. I like drawing. My name is Simon. So if you could have like a freeze frame of DNA as it's starting to get replicated, you would see a whole bunch of these openings where these helicases are coming in and they're just kind of starting in the middle of the zipper and unzipping it from the middle and all over the place. Again, 10,000, up to 10,000, up to 100,000, excuse me, of these per chromosome. So this is happening like all over the place. And these are called replication bubbles. You can kind of see why. They kind of look like a bubble, right? The DNA is opening and it's bubbling out. So these are called replication bubbles. And again, you're going to have hundreds of these, thousands of these, tens of thousands of these on any one DNA strand. So again, it's not really, I mean, it is like a jacket, but also not like a jacket. We're not just doing one zip up and one zip down. We're unzipping it all over the place at the same time. Questions? Cool. One more thing happening. You kind of noticed that too up here. The other thing we have going on is those small circles, the SSBPs, the single-stranded binding proteins are going to bind, they're going to link on to these single strands of DNA, and their job is to keep them open. They don't want them coming back together. We just spent all this time opening them up. We don't want them going back together. We want to keep them open and use these as templates to make some new DNA. Good so far? Cool. Let's move on to what happens next. Oops. Get that backwards in there. Whoops. So 3 prime, shoop, he's going to have a 5 prime end. And the 5 prime, he's down. Okay, there we go. So I know it's backwards from what it was up there, but we got one twist. Because our helicase is moving right along. Up here, he had 2 twists. Now he's just opening more and more DNA, continuing to unzip. New protein coming in. Remember, our DNA builder, which is DNA polymerase, can't start his own chain. Can't do it. So we need this other enzyme called primase. So here's our square enzyme. Primase comes in, and he's going to come in on both strands, the top strand and the bottom strand. I'm choosing a different color. I'm noticing a lot of you guys off camera. Do we need to take a quick break? How are you guys feeling? Quick 5-minute break? Get up, stretch your legs, or just keep on going? Power through? All right. We'll power through. Stay on camera, though, guys. Stay on camera so I can see who's here, who's not here. All right, I got a different color. So if you got, I'll give you a minute here. Grab a, if you have a pen, colored pencil, whatever you have. And if you don't worry about it. You can do this just fine without it. But I'm going to do it to kind of emphasize a little color change here, something different going on. Got these primases. And these primases are making RNA primer. That's why I have a different color here. RNA primer. Now the other big thing is that whole 3' to 5' thing. And I actually need to change mine. I'm sorry. Got confused with the one zip there. I'm going to change these to make this as accurate as possible. Confuse myself, only having one twist. Have to make sure these guys move in the right direction. Remember, they're only adding nucleotides in one direction. 5' to 3', always down, 5 to 3. 5 to 3. I know you might be looking at that and saying, well, wait a minute, you're going 3 to 5. Well, that's the strand that we're reading. The strand that we're building is going to be opposite of that, anti-parallel. So the strand that we're adding to will be 5 to 3. So again, have to make sure it's accurate. Sorry for changing that. Sorry, hopefully none of you guys are writing in pen. Sorry about that. So again, what we've drawn here is primase, that's the square, has landed on each strand, and they build this 10 to 20 RNA. That's kind of key there. These are RNA, that's why we use the green. RNA nucleotides, we call that a primer, like a starter. It gets us started. The whole point of this, of building this primer, is you can remember polymerase can't start his own strand. primase does that for him. All right, now this is key. One of our two primases, this one up here,
Are we just replacing our old ones in the same place?
I think I'm going to.
Because I pulled my files in there, and all the files are from last fall, so maybe either, so I just pulled the whole thing in and didn't, and left that one there. Replication,
Yep,
transcription.
not yet. So primase, one of them just gets to follow right behind. He's moving in the right direction. As this guy, as our helicase continues to unzip more and more of the double helix, this guy just follows right behind him, adding nucleotides. Just kind of works like clockwork. That's pretty easy. The other one, not so easy. See, this guy's moving away from, opposite of the direction that we're unzipping. And he has to, because again, we can only add nucleotides 3' to 5'. He can't add them backwards. He can only add them 3' to 5', which means he has to go away from the direction we're unzipping the DNA. So this leads us to having two strands. They're called the leading strand and the lagging strand. And the reason why they're called that is because if you imagine it to be a race, it's not a race at all, but if you imagine it to be a race, the leading strand's always in the lead. He's following right behind helicase, and he's adding nucleotides pretty much just as they're, as it's being unzipped, he can add new nucleotides. Whereas the lagging strand, you're always going to have this unpaired stretch. It's going to look like he's falling or lagging behind. You have this unpaired stretch of DNA that gets longer and longer as helicase continues to open more and more, until eventually you'll get another primer to come and start another strand. But he always seems to be behind and always seems to be playing catch up. Whereas this guy just follows right behind. So we get a leading strand and a lagging strand, which just falls behind. Questions? You guys ready for that break yet? Doing fine? All right. Get another not much room left. I'm gonna go ahead and finish my last little note. If you guys are out of room, that's fine. No problems with that. Just flip over, start the next page. Not a problem. So quick note on the leading strand. Again, this is the easy one. This is the easy one. Not a problem with this guy. Here's our DNA polymerase, our circle. We got some free nucleotides kind of floating around here, getting ready to get grabbed by this guy and added in there. Nope. All right, so again, one of DNA polymerase's big weaknesses is that it can't start his own strand. That's what we needed primase for. Primase comes in, builds a little primer, a little starting point for him. Then DNA polymerase can come in and start adding on his strands, always in the 5' to 3' direction. And it has to be, that's another one of his weaknesses, it has to go in that direction. So once you have that primer established, that's when your DNA polymerase comes over. That's when you start adding complementary nucleotides. What we mean by complementary is, again, for every A, you add a T. For every C, you add a G. For every G, you add a C. And for every T, you add an A. So that's what we mean by complementary. Questions on the leading strand. Again, he is pretty easy, pretty straightforward, pretty simple. Giving you a quick break here. I'm at the end of a page. I want to make sure everybody gets caught up. And if you have questions, good time to ask them before I flip over. Everybody's good? Ready to flip? Cool. All right. How about the lagging strand? This guy's a little more complicated. Some of the complications we have with the lagging strand is it's constantly having to make new primers. And you kind of envision it. This guy here is going to continue to open up more and more DNA, and it's going to leave more and more exposed DNA that we can only make in that direction, 5' to 3'. I don't know, it says 5' here, but it's because this is 3'. So as helicase continues to open more and more DNA, we're going to have to make another primer. And then another DNA polymerase is going to come over and start building that strand. And this is going to end up resulting on the lagging strand with a whole bunch of different segments of DNA. Because, right, this guy's making his segment, and this guy's making his segment, and this guy's going to make his segment over here. And we're ending up with a whole bunch of different segments, and they're all about 100 to 200 nucleotides long, and they're called Okazaki fragments. They're named after the man that discovered them. It wasn't that long ago, like 30 to 40 years ago. That's like my lifetime, guys. It wasn't that long ago. I believe he was a Japanese scientist. So his name's Okazaki, and he named these fragments after himself, and so that's what we know him as today. So we're starting a new Okazaki fragment right there. This guy's just getting one started. Here's one that's just finishing up, and here's one that goes off the page. And all these different fragments of built DNA. Again, not a problem on the leading strand. The leading strand just makes one continuous strand for quite a ways, because you can just follow right behind helicase. But the lagging strand, we're constantly making new fragments. One special note, you kind of see it right there. And we see it happening right here. Eventually, these DNA polymerases are going to run into the primer of another fragment. And when that happens, they actually pull off those RNA molecules. You can see those getting pulled off, flying away, and it replaces them with DNA. Maddie? How long is your cord? Okay. There is an outlet just inside that door, but I'm not sure you're able to reach it. Check. Okay, do you see like a square in the floor with like wires coming out of it? Go ahead and pull that square up if you can. It's like a lid. And see if, I can't remember if there's an outlet in there or not. No outlet? Okay. What's that? Oh, can you reach? Can you get your cord in there? What's that? Oh, then probably close it. Go ahead and shut it. I didn't realize there were bugs in there. That's disgusting. Oh, like a lot of bugs? Leave it open so they get scared by the light. Hopefully they'll. Oh. Actually, I don't know what's worse, dead bugs or living bugs. You've handled that very well. Getting some juice? All right. People at home, you guys missed some good entertainment here. We'll talk about it someday. All right. How we doing? Good on logging strand? Questions? We're almost there, guys. Almost there. Got one more step to make. Doing well? Let's finish these bad boys. Cool. Okay.
I'm starting to go off the bottom.
Oops. Sorry about that. Thank you. Sorry about that. How long were you out? Sorry about that. Are you getting juice? Is your computer going up? Okay. All right. So to finish up here, eventually, and it doesn't matter if it's a leading strand or the lagging strand, eventually DNA polymerase is going to run into the back end of another strand. It's going to run into the primer. It's going to remove the primer. It's going to start building DNA, but eventually it's going to catch up to that next strand, and it won't be able to connect those two strands. Now, this happens a lot more often in the lagging strand. Think about all those Okazaki fragments. So it's going to happen a lot more often in the lagging strand, but it does still happen in the leading strand, just not nearly as often. So eventually it gets to the point where, okay, we've got all the strands, but we can't link all the strands together. We've got this gap right here. The reason why? DNA polymerase can't connect 3' to 5'. It just can't do it. So to kind of put the final finishing touches to build that link right there, that's where we get our final enzyme involved. Ligase. So that's where a final player comes in, ligase, and his sole job is to link up these segments. We're going to have a lot of these segments, especially in the lagging strand. We're going to have some on the leading strand, but we're going to have tons of these little gaps on the lagging strand. And so it's the ligase's job. His sole job is to come in and make that final 3' to 5' link. Questions? Okay, let's wrap this up. Just a couple notes here at the end to finalize this whole thing. We're just about there. Promise, 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 promise. Oops. Proofreading. Humus. It's a bone, I think. Humans. This process happens fast, super fast. DNA polymerase is adding nucleotides at a rate of 50 per second. I mean, you guys know how long a second is. Like that. 50 nucleotides right there. 50 A's, T's, C's, G's, links in there. Done in 1 second. So that is super, super, super fast. When you're moving that fast, you're going to make mistakes. When think about how fast you guys type. We talk about words per minute. If I gave you a keyboard and you got 4 keys to hit, A, T, C, and G, and you need to hit 50 of them every second. First of all, I don't think any of us could. But even if you could hit that, how many mistakes might you make? You're going to have a few mistakes in there. DNA polymerase makes 1 in 10,000 nucleotides, which is pretty good. Again, think about it. I'm going to give you 4 keys you got to hit: T, C, G, and A. How many mistakes would you make if you're trying to hit 50 keys per second? You're going to make a lot more than 1 in 10,000. That's fine. We're human. DNA polymerase, 1 in 10,000. But he fixes them. Well, at least his cousin, another DNA polymerase, fixes them. So even after replication has completed, you've got your 2 strands of DNA. Those DNA polymerases continue to kind of go up and down the strand and make sure that all the A's are paired with T's. All the T's are paired with A's, all the C's are paired with G's, and all the G's are paired with C's. They're proofreading. With proofreading, we get that 1 in 10,000 down to 1 in a billion. That's a billion right there. Enough zeros to be a billion. So we really get that mistake rate to a very smaller, much smaller number. Now, mistakes still happen, mutations still happen. We'll talk about those a little bit more next week. But we really get those down. Final note, last note. This is it. Here we go. Pencil down. Whew. Did it. So I survived another week of biology. How about that? So we're all pretty well aware. We talked about this earlier in the unit, or last unit, I should say. DNA replication takes place during S phase. That's what the S stands for, synthesis. In humans, it takes 1 hour. That's it. You can copy your entire genomic library in 1 hour. That's incredibly impressive. We were talking yesterday how individual chromosomes have 50 million to 300 million nucleotides. That's one chromosome. You've got 46 of them. So to think that we could copy 46 times 200 million in 1 hour, that's That's amazing. It's amazing. Questions on the notes? Anybody need to see anything, you missed parts of a page or something? How are those hands? Need to shake them out? At least it's entertaining stuff though, right? Really cool and fascinating. Piqued your curiosity. Okay. All right. Tonight, you guys have a homework due. It was from Tuesday. You guys were here last on Tuesday. So Tuesday's homework, if for whatever reason you haven't done it yet, you got to get that done. It's due tonight. There's a new homework now posted over this material, the stuff we just looked at today. So the new homework is due tomorrow night. It's one of those activities you guys like, the online ones. You guys told me you like those. So you get unlimited attempts and get your perfect score if you want to go after that. So it's over the stuff we just looked at. It's on Canvas. You can take a look at that and get that done by tomorrow night. If you have questions with it, you have a problem with it, tomorrow I've got